is Abigail McLennan, and I will be speaking about the latest research update from the Surveillance of Equine Strangles project, titled Utilising Genomics to Identify Potential Streptococcus equi contact networks in the United Kingdom. I'd like to thank the Horse Trust and SEIB insurance brokers for funding this project, all of the participating laboratories within the SES network that contributed data to this analysis, as well as the Sanger Institute for facilitating whole genome sequencing. Strangles is an infectious respiratory disease of horses caused by the bacteria Streptococcus equi and is considered endemic amongst the UK horse population. Until 2018, when the Surveillance of Equine Strangles project was launched, there was no dedicated national surveillance data monitoring the extent of strangles across the UK. SES combines the collection of epidemiological data relating to laboratory-confirmed strangles diagnoses with the submission of bacterial isolates to enable analysis into outbreaks at an unprecedented level. This information can then be used to investigate the potential transmission of S. equi amongst horses infected with a genetically identical strain through molecular and contact network analysis. This branch of research has been named sociomolecular epidemiology and has been developing within the last decade. Whole genome sequencing and molecular analysis provides an in-depth look at the circulating strains of a pathogen amongst a group of sampled infected individuals, whereas contact network analysis often gathers more descriptive data regarding the network of individuals being investigated. This is due to enrolled members, which can be either infected or not infected, often giving details about unenrolled members of the network, providing a wider understanding of group dynamics. When linking this information together, better insights can be gained of the dynamics between those infected and sampled, and therefore included in whole genome sequencing analysis, and those not sampled or non-infected who may have played a part in transmission throughout the network. In relation to strangles investigation, potential transmission events can be identified using whole genome sequencing data. Then, when linked with contact network analysis, data from a wider sample of those affected but not necessarily tested, new insights into the transmission of strangles could be revealed in new levels of detail previously not recorded before. The study objectives were to utilise whole genome sequencing data available for S. equi isolates to identify related strain types with accompanying epidemiological data reported to SES to help direct future contact network analysis. 348 S. equi isolates from freezer archives and isolates submitted to SES from 4 UK veterinary diagnostic laboratories between 2016 and 2019 underwent whole genome sequencing. Analysis of whole genome sequencing data was undertaken using Pathogen Watch, where assembled data was imported and isolate strains were compared, and a phylogenetic tree reconstructed. Isolate population structure was inferred using BAP software, which categorized isolates into pre-classified S. equi pathogen watch clusters. The phylogenetic tree and associated epidemiological metadata was then visualized in MicroReact to view groups of isolates that clustered together on the tree. These clusters of isolates were then confirmed as genetically identical using Pathogen Watch's score matrix, where a score of 0 indicates no differences in single nucleotide polymorphisms between isolates. Our results show that S. equi strains have continued to evolve since the last published analyses. Previously, the majority of S. equi strains were associated with the more ancient strain type of Pathogen Watch 5, shown in red on the bar to the right of the screen. However, these results now show the majority of strains sampled in the UK between 2016 and 2019 are associated with the recently emerged Pathogen Watch 2 strain type, shown in blue on the bar to the right. Previous research in 2011 identified a slight shift in the prevalence of Pathogen Watch 5 isolates compared to Pathogen Watch 2. Therefore, our results suggest that there has indeed been a change in the circulating strains of S. equi in the field within the last decade, where Pathogen Watch 2 is now the dominant cluster of S. equi identified in the United Kingdom. This finding is important as the currently available vaccines and diagnostic tests for strangles were developed for Pathogen Watch 5 strains. Phylogenetic analysis from the 348 isolate sequence revealed 33 distinct groups that were genetically identical. I will now give an example of a group of isolates where through whole genome sequencing and epidemiological data gathered for SES, we can infer that there may have been a possible transmission event which warrants further investigation through contact network analysis. 5 samples were diagnosed by 3 labs reporting to SES, which were found to be genetically identical. These 5 samples are from 4 horses and were submitted by 4 veterinary practices from 4 different regions of the UK over a 90-day period in 2019. Some epidemiological data is available for each sample, but further investigation using contact network analysis is required to establish if these horses are in fact linked. Amongst the archived isolates sequenced, 1 sample was identified to be genetically identical to the Equilis streptococcus vaccine strain. This sample is part of the Pathogen Watch 5 cluster of isolates and is circled in teal at the top of the phylogenetic tree. Via additional information gained through contacting the submitting veterinary surgeon, it was found that this sample came from a thoroughbred colt recently transported from Ireland to Singapore. The colt was vaccinated submucosally into the inside of the upper lip with Equilis streptococcus on 2 occasions a month apart prior to travelling. The horse subsequently developed an abscess on the neck a month after the second vaccination, which was sampled and confirmed as S. equi. There are a few limitations when utilising molecular epidemiology. Often this branch of research is used as a retrospective analysis tool, meaning that there is a lag time between the sampling events and the sequencing of these isolates. As whole genome sequencing technologies become more accessible, it is hoped that this lag time can be reduced, allowing for a quicker analysis and faster delivery advice to help control an outbreak. And as found previously when analysing descriptive data gathered via SES, there is often incomplete or unreported clinical information provided alongside a sample when it is submitted for diagnostic testing. This lack of detailed epidemiological data limits the ability to gain a comprehensive understanding of the potentially linked transmission events, reducing the effectiveness of molecular epidemiology. Our findings have shown that through whole genome sequencing and phylogenetic analysis, 33 distinct groups of genetically identical S. equi isolates were identified from 348 samples collected between 2016 and 2019. Through the basic epidemiological data submitted with these isolates, inferences can be made that these horses may have been infected through transmission events such as attending a competition and mixing with unknown horses, or introducing a new horse to a yard without following advised quarantine and screening protocols. These groups of identical isolates warrant further investigation through the use of contact network analysis to determine if there was in fact an epidemiological link between infected horses within each group. Our data shows that the whole genome sequencing of bacterial isolates and phylogenetic analysis, visualized with accompanying epidemiological metadata, is an important step in the understanding of strangles transmission amongst groups of horses. The movement of horses throughout the equine industry can be local, national, or international, and this new epidemiological approach is not just limited to UK isolates, but can be utilised for S. equi isolates collected worldwide to determine transmission events that may have occurred on an international scale. Thank you for watching, and I will gladly answer any questions that you may have.